cells, tissues, and organs. So various organs are built by different tissues, and when we look at this cellular level, we see striking differences there. So here you have connective tissue cell, bone cell, skin cell, muscle, nerve, gland, red blood cell, and white blood. But all these cells originate from identical genome, right? This genetic information is the same. Yet they differentiate into these tissues and organs. So how does it work? So we know that this is about gene regulation, right? And we talked about gene regulation a little bit yesterday, and you have some knowledge from biochem and other sources. So the major differentiation like that during development is achieved through cascades of regulatory changes, driven mostly by regulatory proteins known as transcription factors, right? So we saw yesterday that promoter region with a complex of various proteins, including transcription factors, bind to the promoter region. And there's some other regulatory proteins like enhancers and repressors. And then epigenetics was discovered. Genetics is genetic variation that is not caused by mutations at DNA level. There were some chemical modifications to nucleic acids, to histones, and we're gonna talk in detail about these mechanisms and pathways. So then people jumped on saying that this differentiation that we see is actually epigenetics, that all we see here is due to epigenetics. So this is the broad kind of perception of epigenetics, and some people are still using this term in this context. But that's wrong, because this is not the epigenetics. So like I said, this is something that we knew quite a while back. It was pretty well determined that these processes are driven by transcription factors and regulatory changes. So yes, epigenetics is a part of regulation, gene regulation, very important, but overall, we don't really need epigenetics to explain this differentiation here. So what is epigenetics then? This is the definition that we're going to use. Epigenetics is the study of heritable changes in cellular function or gene expression that can be transmitted from cell to cell and even generation to generation, and that's the most controversial part of it, but it happens as a result of chromatin-based molecular signals rather than mutations at DNA level. So that changes chromatin structure. We talked about euchromatin, which stains lighter, and those are regions within nucleus that are transcriptional active. So these regions that stain lighter contain are rich in genes, and these genes tend to be transcriptionally active. Versus heterochromatin, those are the darker parts of the nucleus. This is heterochromatin. And for the most part, heterochromatin is it contains repeat sequences. It's poor in terms of gene contents. It may contain genes, but these genes are typically silenced. So these parts of chromatin are transcriptionally silent. This slide summarizes major pathways of epigenetic regulation. So, like I said, when I showed you the definition, epigenetic is about reconfigurations, reprogramming of the chromatin. It's about the chromatin structure, right? But these changes in chromatin structure are chemical, right? So we need to look at the pathways, or the biochemical pathways that drive epigenetic reconfigurations of chromatin. So we have chromosome, and then you have nucleosomes and histones with these protruding histone tails. So those are N-terminal tails of these proteins. And of course you have DNA. It winds around the histones. So those are the major sources of epigenetic variation. The first one, we can talk about them as pathways. The first one is modifications of histone tails. Then histone variants. So it turns out that different parts of chromatin may recruit different types of histones. And more on this in a second. And finally, DNA methylation. This is probably the mechanism that we know most about. So again, three major sources of epigenetic regulation. So let's start with histone variants that are characteristic to various parts of chromatin. So you know that when you look at nucleosome, we have an octamer of histones, right? We have a linker H1 histone. This is the yellow part, but the core consists of four types of histones: H2A, H2B, H3, and H4, and they are present in duplicates, forming the histone octamer. But turns out that these histones are encoded by multiple genes. And first we thought that these genes are just there for redundancy. And we thought that these duplicates are identical by sequence, forming these histones. But then when we compared sequences, we saw differences there. So the first histones within each major family, like H2A, were divided into subfamilies. Again, based on some variation that we see, even though clearly each of them is H2A, there are some differences in sequence that turns out translate into some structural changes as well, and functional. So function comes with structure. So there was some hidden level of variation and function variation that we were not aware of. So then researchers started to compile all this information about all these genes, this genic variation, which we call members. So members are individual genes. They can be still present in duplicates or multiple copies, but these members are differentiated based on sequence variation. So turns out again that there is a level of variation that we first were not aware of. It doesn't mean that it changes the number of histones within the nucleosome. It does not. It's still two H2A, two H2B, two H3, and H4. But various members may go may fall under this category. So we still don't quite understand how the two shapes, how these various members, these histone variants, change the structure of chromatin. But we have some good examples and understanding. So this is like a typical nucleosome, and this is centromeric nucleosome, SENPA. This is again, this is a nucleosome that occurs in the centromeric regions of chromosomes. We know that these centromeres consist mostly of heterochromatin. It's highly packed, very compact type of heterochromatin there. And indeed, the nucleosome structure, when we look at that, is different due to different differential recruitment of histone proteins. That make sense? Mm-hmm. Say that again. We're comparing euchromatin and heterochromatin. More specifically, this is heterochromatin from centromere. Again, centromere are highly compact, and this is achieved through the structure of SENPA. So this is nucleosome that occurs in centromeres of chromosomes. So this is still an octamer of histones, H2A, H2B, H3, and H4, but different members compared with regular euchromatin nucleosome. And then histones can be chemically modified. So this is a major class of epigenetic signaling consisting of various modifications to any of the core histone types. Again, that applies to H2A, H2B, H3, and H4. And such modifications include histone methylation, phosphorylation, acetylation, and some other less frequent chemical modifications. So these modifications are located typically on the N-terminal tails of histones. So you can see the tails here, and this is zoomed in. So you see specific, you see individual amino acids in the tail. So let's start. So this is methylation. The black dots correspond to methylation. The methylation effect depends on the position, on very particular position of amino acids. So these modifications may either suppress expression, or they may increase expression from the region. So these modifications, when I say suppress, it means that such a modification makes chromatin more compact. It just closes, and it's less accessible to translational machinery and transcriptional machinery. when I say that it increases expression, it means that it opens up chromatin, that it makes it more like euchromatin, so it's transcriptionally active. So like this position 4, this K corresponds to lysine. So this is lysine number 4 in this H3 N-tail, H3 histone. And here's a trimethylation. This trimethylation signal causes derepression and increased expression of genes. But this is different. I told you that methylation marks, even on the same tail, may have conflicting opposite effects, because this lysine 9, when it's dimethylated, it actually causes silencing of the region. So you see that we have multiple methylation marks, and it's like a tricky balance between suppression and derepression instructions for chromatin. Mm-hmm.
Is there a way for us to tell by looking at this picture whether methylation is activating or
Not really. So you need to know that. But we have a pretty good understanding of these marks. It's really a lot to look at, because these signals may also differ between tissues. So they may give different instructions in different tissues to complicate things even more. But the bottom line is that we get better understanding of these methylation marks. They need to be mapped again with respect to tissues and individual histones and positions. We have an array of methods to use, but it's quite tedious actually. So typically we have antibodies that are specific to each modification in each sequence of amino acids. So the antibodies can recognize very specific targets with very specific methylation or acetylation patterns. So that was methylation that as I said, it's not consistent, so we cannot really tell if this is up or down regulation, because these effects depend on the location. This is in contrast to DNA acetylation. And turns out that acetylation removes the positive charge on the histones, decreasing the interaction of the N-termini of histones with the negatively charged phosphate groups of DNA. As a consequence, the condensed chromatin is transformed into more relaxed structure that is transcriptionally active and associated with higher levels of gene transcription. This relaxation, or acetylation in general, is conveyed through this HAT enzyme. This is histone acetylate transferase enzyme that is adding these acetylate groups to N-termini. This process can be reversed through HDAC, which is histone deacetylase, and by doing so, and chromatin closes up to form heterochromatin. And again, heterochromatin, this closed chromatin is transcriptionally silent versus relaxed chromatin or euchromatin that is transcriptionally active. Now that we have recognized these players, this enzyme, we can actually target them using some inhibitors, chemical inhibitors, and modify or control acetylation in histones in an experimental way. So there are some drugs based on these inhibitors. So in a sense, those are like epigenetic drugs. And finally, DNA methylation, which is, like I said, the best recognized mechanism, epigenetic mechanism and pathway. It's relatively simple and allows us to provide methods that, or we have methods that enable us to see these methylation patterns of DNA. So this is about cytosine methylation. There is the methyl group that is added to the 5th position of the pyrimidine ring. And this is conducted by DNMT enzyme, which is DNA methyltransferase, which transforms cytosine into 5-methylcytosine. Typically, DNA methylation occurs on the C of CpG dinucleotides. We call these CpG sites CpG islands. So that's, again, this C tends to be followed by G at the same site. If C occurs among other nucleotides, it's less frequently methylated. And the most important thing probably is that methylation leads to inactivation or silencing of a gene. So when it occurs in like a promoter region, this is something that directly suppresses expression from this region, suppresses expression from the gene. If there is no methylation in the region, the gene is active. This is a tricky part because when I say that methylation silences a gene, inactivates it, you have to remember that sometimes we have negative feedback interactions, right? So sometimes one gene is silenced, but that because this gene regulates some other gene, or regulates this protein regulates some other protein, then the other protein may actually increase in expression, right? But this is like a further down effect. The direct effect is this silencing and downregulation of the target gene. This mechanism is also prevalent in genomic regions that contain transposable elements. So transposable elements tend to be methylated because this is kind of, you can think of it as a genomic defense system against transposable elements through their methylation. So when transposable elements are methylated, they tend to be silent and they don't transpose from one side to another. So this line shows you maintenance of methylation through DNA replications, because when DNA replicates, there is the new copy of DNA doesn't inherit methylation marks in itself. So again, if there are no methyltransferases present, the methylation marks that were there at DNA level are lost. So it's like kind of passive loss of DNA methylation marks, or the cytosine methylation. But there is a DNMT1 enzyme, DNA methylase transferase 1, that is responsible for copying DNA methylation marks to daughter strands during DNA replication. So this once a new strand is formed, this DNMT1 senses marks on the template strand and copies it on the new daughter strand. X chromosome activation that I mentioned before several times is probably the most dramatic epigenetic effect that we see in cells, because it's not just a single gene silencing, it's the whole chromosome that is shut down through epigenetic mechanisms. So the whole process starts with this Zik region, and this region of the X chromosome contains ZIST gene, which is very interesting gene because it doesn't really encode a protein, it encodes a long non-coding RNA. So this RNA is never translated into a protein. But even though it's not translated, it's still functional. It regulates epigenetic pathways leading to this suppression of X chromosome. So it spreads from the region, from the Zik region, downstream and upstream along the X chromosome, leading to complete inactivation of X chromosome. And then this X chromosome forms this tiny structure known as Barr's body, right? It's something that we can see easily under the microscope, that only one X chromosome is large and active, and the other shrinks into this Barr's body. So, in other words, there are other players, other epigenetic players than those that I mentioned, other than DNA methylation, histone modifications, and histone unit variation, namely non-coding RNAs. So this is an example. This is just an example of non-coding RNAs. Like there are many microRNAs, very small non-coding RNAs that also are involved in these epigenetic pathways, interact with proteins and other RNAs to exert these silencing effects. So now I wanted to tell you about imprinting of genes. This is something that happens in egg and sperm environment, because these cells are very different. The germline, to start with, is a very different environment for cells, and sperm has to function as sperm, of course, and eggs have their own unique functions. There's some unique transcription that occurs in egg, and it's different from that happens in sperm. So that leads to some epigenetic changes, some epigenetic reprogramming that marks chromosomes from father differently from those that are in mother. So in other words, there's differential epigenetic silencing in eggs and sperm. But that also creates some problems after fertilization. So when the zygote is formed, these chromosomes have some specific patterning epigenetic marks that need to be erased during development, and then when different tissues differentiate, they also create very unique environments requiring unique ways of regulation, including epigenetic regulation. So then genes acquire new epigenetic marks through embryogenesis and development. So first what we see that we have these maternal and paternal marks. They land in the zygote after fertilization, and then we see the first phase or the first wave of demethylation. So all these marks, not all of them, as you will see in a moment, but most of these marks are erased. The mechanism is most likely passive DNA demethylation. So there's no active, there's no enzymes that do that. It's just DNA replication, and as I told you, through DNA replication, these methylation marks tend to be lost over the subsequent cellular divisions. Then in the embryogenesis, new imprints and epigenetic marks show up, and then we see the germline, right? This is the germline where germ cells or gametes are formed. So the next slide will show you that better, but these marks now that come from development have to be erased again. So this is the second wave of demethylation. And it's quite rapid, so there's some active mechanism of demethylation involved, which we still try to understand how that works. But no matter what it is, erasure of imprints takes place. So these chromosomes now may receive new maternal and paternal marks, dependent on whether they are in eggs or sperm. So again, from this slide, what you want to remember is that there are two major ways of demethylation or epigenetic reprogramming in general. It's right after the zygote formation in early embryogenesis, and then in primordial germ cells in the germline, leading to gametes, and gametes, like I said, receive unique imprints based on the gender. So this slide shows you why this demethylation is important, this reprogramming is important. Because somatic cells have these maternal and paternal imprints. So let's say that that now this blue one is maternal, but if this is a female, these marks have to be reprogrammed. So now this blue chromosome, even though it was paternal, now it ends up in an egg, so it has to receive maternal, it has to receive female marks. So this is the reprogramming that happens in gametes, in female gametes. And conversely, this maternal chromosome, or the maternal chromosome still carries some marks, maternal marks, has to be reprogrammed. These marks have to be raised in sperm cells and receive the male specific marks. Yes. Yes, so when I talk about that, I mean that some imprints avoid erasure. So like when I told you that these marks, these maternal and paternal marks are lost, it turns out that not all of them are lost. Even more so, it's important that they persist. So there are some genes, it's around 100 of them in humans, that have these maternal or paternal marks. And it's important that the mark stays there as is, that it's persistent, because if it's lost or if it's reversed from a male to female, or the other way around, it leads to some epigenetic diseases, and I'm going to talk about them in a moment. So, I'm sorry? Marks is like methylation pattern, right? So there's some methylation mark specific for maternal chromosomes. There are some methylation marks specific for paternal chromosomes. So most of them are lost here, but there are some persistent marks. Like I said, around 100 genes that differentially silence genes based on the imprint. Imprint means that typically this gene is silent. So if there's maternal imprint, this gene is silenced and only paternal copy is active. So it's important, this kind of chromosomal memory is important for some genes. If this memory is affected or reversed, it leads to some dysfunctions. But all these marks, again, even those most persistent marks, have to be reprogrammed in eggs and sperm cells. So now, like I said, even though there were some maternal marks here, all these maternal marks now have to be reversed into male marks in sperm cells. Does that make sense? It's quite complex, and the next slides will be actually the most difficult slides that I have for you, I think, in this course. So please bear with me if you have any questions, let me know. So this is probably the most famous epigenetic disease that occurs as a result of changes in epigenetic programming. But turns out that the most common change that actually affects the epigenetics is deletion. It's a deletion of a fragment of chromosome 15. It's 15q11. Between 15q11 and q13, there's a region that is enriched in these maternally and paternally imprinted genes And it's important that this pattern is not affected. If there is an event, a genetic event like deletion, it changes the epigenetic balance and it leads to either Prader-Willi or Angelman syndrome. So both syndromes, Prader-Willi and Angelman, result from the same deletion. But the phenotype expression is dependent on the parent, which parent contributed the deleted chromosome. So if this is Prader-Willi syndrome, this is the paternal chromosome that contributed. So we expect, so not we, the expression is expected in the region from maternal, from paternal genes. And if these genes, if expression from these genes is missing, this is leading to Prader-Willi syndrome. In Angelman syndrome, this is caused by maternal chromosome. So expression from maternal genes is expected in this region. When it's missing, for example, due to deletion in this region, that leads to Angelman syndrome. This is also known as parent of origin effect. So we have genomic imprinting and parent of origin. Parent of origin is like a very broad term that refers to something that, again, effect depends on whether it's due to maternal or paternal contribution. I know that you are probably confused, but this slide gives you more information, and the next slide gives you even more information. So I'm going to build on what I just said. So we're going to beat this to death, if you like. So this is Angelman and Prader-Willi syndromes, and we have this 15q1113 region. And now this blue color corresponds to expression from paternal genes. The red color is expression from maternal genes. So you can see that this region contains many genes, right? And like for example, this MAGO2 gene, you can see that is in blue. So this is paternal expression from the paternal copy, and these maternal copies of these genes that are dotted is silenced. So if it's blue, again, it means that it's expression from the paternal copy, which in other words means that this is maternal imprint. Maternal imprint means that the maternal copy is imprinted or silenced, and that's why only paternal copy is expressed. And like here, you have this UBE3A gene. I don't know if you can see it. It's the red box, and there is no blue box, which means that this is maternally expressed now, and the paternal copy is silenced. If it's red, again, it's maternally expressed, which means that there is a paternal imprint now, because the paternal copy is imprinted or silenced. And that's mostly due to differential methylation of the region. Okay, so those are the persistent. So actually, this is so this pattern that you see here, these blue and red boxes, is the normal situation. This is what we expect in normal individuals. If it's both red and blue, it means that expression is from both copies. It's a biallelic expression. Okay, so there are some genes in this region that are expressed from both copies, There are some genes in this region that are expressed only from maternal genes, and there are some that are expressed only from paternal genes. Okay? And this pattern has to be preserved for normal functioning. So those are like these persistent imprint signals that I mentioned, that they have to be there for normal function. Now, if something happens to these maternal additions, to these genes that are expressed maternally, those blue ones, then we see Angelman syndrome. If the syndrome, if genes now, those in blue boxes, are affected, for example, they're missing, or they're reprogrammed in a different way, that leads to PWS, this Prader-Willi syndrome. And you can see that even though this is the same genomic region, these syndromes differ a lot even in infancy. So like there's quite striking differences there. Prolonged suckling in Angelman, poor suckling in Prader-Willi, frequent crying in Angelman, weak crying in PWS, hyperactive, sleepless, inactive, sleepy, microcephalic, dolichocephalic, tend to autism spectrum, tend to psychotic spectrum disorder. So very striking differences there. Again, due to genetic or epigenetic changes in the same region. And this is a very busy slide, so maybe let's do it after the break. This slide also explains the mechanism, and I think that